Hello everyone, you're welcome on my YouTube channel Capsule. This is Dr. Vineet Kumar. I will tell you about the pH partition theory. This is the trailing video of my previous video on the theories of drug dissolution. As far as the pH partition theory is concerned, it is related to the absorption of the ionic drug into the systemic circulation. The absorption of the ionic drug depends on the pKa value of the drug as well as the pH of the site at which or from where the drug is being absorbed into the systemic circulation. So here the pH partition theory explained the process of drug absorption from the GI tract in simple terms. As per this theory, the process of absorption of the ionic drug that primary permeate by the passive diffusion and has molecular weight less than 100 Dalton is governed by the drug's dissociation constant, that is pKa value, lipid solubility of the drug or unionized form of the drug, that is log P value, and pH at the absorption site. So the absorption of these kind of drugs will be based on these three factors, that is pK, log P and pH of the absorption site. Since most of the drugs are weak electrolytes like weak acid or weak base, their degree of ionization depends upon the pH of the biological fluid. If the pH on either side of the membrane is different, then the absorption will be different. So if the pH on either side of the membrane is different, then the compartment whose pH favors greater ionization will contain greater amount of the drug. Greater ionization means the side which is favoring higher ionization will have the greater amount of the drug. The drug will not absorb to the other side. Only the unionized fraction of the drug will be absorbed if sufficiently lipid soluble. And the passive diffusion is seen until the equilibrium is attained. So when the fraction of unionized drug on both the sides will be equal, then the diffusion will be stopped. However, the above statement of the hypothesis was based on the assumptions that the GI tract is a simple lipoidal barrier that transport to the transport of the drug. The GI tract is considered as a simple lipoidal barrier. The other factors related to these GI tract is not considered in this theory. Larger the fraction of the unionized drug, faster will be the absorption rate. And the greater the lipophilicity of the drug, of the unionized form of the drug, better will be the absorption. So all these factors are considered, but these are associated with some limitations also. And this theory fixes these parameters that the biological membrane, the GI tract is simple lipoidal biological membrane, and only unionized drug will be absorbed, and the lipophilicity will be should be high for better absorption. So these can be considered as constant as well as limitations also. In this slide, I'll explain about the drug pK and gastrointestinal pH and the relation of the absorption of the drug with the pK value and gastrointestinal pH. So the amount of the drug that exists in unionized form is based on the dissociation constant of the drug and pH of the fluid at the absorption site. So these are the two factors that affect the absorption of the unionized form of the drug. So the pK value will be affected by the pH at the absorption site. As this theory states, the dissociation constant of both acidic and drugs are expressed as pK value. So whether the drug is acidic or basic, then we will use only pK value, pK as a term of expression. Now, if the drug is acidic, then what will happen? So in the case of acidic drug, the lower pK value means stronger acid. And if the drug is stronger acid, then the greater the proportion of the ionized form of the drug at particular pH, then its ionization will be high. So if the drug is basic, then higher pK value means the stronger base and greater the absorption of the ionized form of the drug at particular pH. So the lower pK value for acidic drug and higher pK value for basic drug will highly affect the absorption of these kind of drug into the systemic circulation. Thus, from the knowledge of pK of the drug and pH at the absorption site, the relative amount of ionized and unionized drug in solution at particular pH and the percent of drug ionized at this pH can be determined by Henderson-Hasselbalch equation. So from the previous discussion, we can see that the stronger acid or stronger base will not be absorbed efficiently, and for better absorption, the drug should be a weak acid or weak base. So the Henderson-Hasselbalch equation will be true for weak acid and weak bases. So for weak acid, the Henderson-Hasselbalch equation will be the pH equal to pK plus log ionized drug upon unionized drug. The percent of drug ionization will be determined by this equation, that is equal to 10 to the power pH minus pK upon 1 plus 10 to the power pH minus pK into 100. And for weak bases, the equation will be pH equal to pK plus log, that is unionized drug upon ionized drug, and the percent drug ionized will be calculated by the equation 10 to the power pK minus pH upon 1 plus 10 to the power pK minus pH into 100. So these are the Henderson-Hasselbalch equation for weak acids and weak bases. So when the concentration of ionized and unionized drug become equal, the second term of the equation 1 and 2. So here I'm talking about the second term, this and this. So if the concentration of ionized drug and unionized drug become equal, so if these terms are equal, then these overall term will be 1, and as we all know that log 1 is 0, then in that condition this equation will be reduced to 0. This term will be reduced to 0 and pH will be equal to pK value. So this is true for both weak acid and weak bases. This term will be equal to 1. Here also the term will be equal to 1. So this is the simplified Henderson-Hasselbalch equation. Now the pK is a characteristic of the drug. Just like log P, it is also a characteristic of the drug. So if there is membrane barrier that separates the aqueous solution of the different pH, then the theoretical ratio R of the drug concentration on the either side of the membrane can be given by the equation derived from Soar et al. This is the equation for weak acid, and here you can see that Ra, that is theoretical ratio of the drug concentration into GI tract and plasma, and that is equal to 1 plus 10 to the power pH GIT minus pK upon 1 plus 10 to the power pH plasma minus pK. So for weak bases, the equation will be that is Rb equal to C GIT upon C plasma, that is equal to 1 plus 10 to the power pK minus P GIT upon 1 plus 10 to the power pK minus pH plasma. So these are the equations derived from Soar et al. For determining the theoretical ratio of the drug concentration on either side of the membrane. So by these equations, one can calculate the relative amount of unionized and ionized drug form of the drug and predict the extent of absorption at given pH of GI tract. Now, considering the pH of gastrointestinal tract in the range of 1 to 8, that is normally considered. The pH of the stomach is 1.5 or somewhere around 2, and the pH of intestine maximum up to 8. So we can consider this pH range from 1 to 8. And if we consider this range 1 to 8, then certain generalization can be made for the absorption of drugs as per the pH partition theory. So here for weak acids, if the drug is very weak acid, so its pK value will be more than 8. And the examples of such acids are phenytoin, ethoxyamide, and several barbiturates. So if the drug is weak acid, then these are essentially unionized at the all pH conditions of the gastrointestinal tract, and their absorption is rapid and independent of gastrointestinal pH. So this is true in the case of weak acid, as we have already discussed. Now, if the acid is having the pKa range from 2.5 to 7.5, that is moderately acid. So some of the examples of this kind of drug are aspirin, ibuprofen, phenylbutazone, penicillin analogs, etc., etc. So these drugs are greatly affected by the change in the pH of the gastrointestinal tract, and therefore their absorption is pH dependent. So as the pH changes, their absorption will be different at the different location. Such drugs are better absorbed from the acidic condition, that is where the pH is less than the pK value. So these drugs will be absorbed in the acidic condition, where they largely exist in unionized form. Note, for the better absorption of the drug should be in the unionized form. If the drug is in ionized form, then its absorption will be low. Now for stronger acid, which is having a pK value less than 2.5, as we have already discussed, the example is chromoline sodium. It is ionized in the entire pH range of the gastrointestinal tract, and therefore remain poorly absorbed. So it is stronger acid, as we have already discussed. The stronger acid will be absorbed less as compared to the weak acids. So this is true in this case. Now, if the drug is basic, for a weak basis, the pK value will be less than 5. The suitable examples of this category are caffeine, theophylline, diazepam, and oxazepam. So these are essentially unionized at all pH values. If they are unionized at all pH value, their absorption will be rapid and will be good at all pH conditions. So if the drug is moderately basic, that is range from 5 to 11, the examples are morphine, chloroquine, empyramine, and amitriptyline like drugs. These will be absorbed from the relatively alkaline conditions of the intestine. So these will be absorbed better from the intestine. And if these are the stronger bases, that is pK more than 11, examples are mecamylamine and guanethidine. These are ionized at all pH condition of the gastrointestinal tract, and therefore these drugs will be poorly absorbed from the gastrointestinal tract. So this is the list of the drugs. We can see that influence of drug pK and gastrointestinal pH on drug absorption we can see here. And in the case of phenobarbital, that is in the category of weak acids, and these drugs are in the category of weak acid, the pK value is more than 7, you can see here. So these drugs will be unionized at all pH values, and if they are unionized at all pH value, their absorption will be good from the entire length of the gastrointestinal tract. The examples are phenobarbital, hexabarbital, phenytoin, ethoxyamide, and so on. So in the case of moderately weak acids, the pK value are less and or you can say moderate, so they will be unionized at gastric pH, lower pH, and ionized in intestinal pH. At higher pH, these will be ionized, and therefore these drugs are absorbed from stomach only. So in the case of a stronger acid, these are having the pK value less than 2 or 2, and so this will be ionized at the entire length of the gastrointestinal tract and at all the pH. So these will be the poorly absorbed drug from the gastrointestinal tract. Now we will see the influence of drug pKa and gastrointestinal tract and of drug absorption of weak bases. So bases. So if we talk about the weak bases, then the examples are theophylline, caffeine, oxygiform, diaziform, and these are having the pK value very less. So this will be unionized at all the pH and absorbed the good amount from the gastrointestinal tract. And if they are moderately weak bases, then the examples are reserpine, heroin, codeine, and amitriptyline. So these will be ionized at gastric pH and relatively unionized at intestinal pH. So these drugs will be absorbed better from the intestine. And in the case of stronger bases, this will not be absorbed from the gastrointestinal tract, and the examples are melamine and guanethidine. Now we will see the influence of pH on ionization of the drug. We have some denotions like Ha, BOH. These are termed as the concentration of unionized acid and bases respectively, and A minus and B plus are the concentration of ionized acid and bases respectively. So we will see how the pH of the gastrointestinal tract will affect the absorption of the drugs. Suppose these are the membranes, and these membranes separate the stomach from the plasma, and then intestine from the plasma. So in the first case, or you can say the first example, weak acid, that is ibuprofen, pK value is 4.4 here. It is in the stomach, it is having the more concentration of the unionized form and less concentration of the ionized form. So it will better absorb from the stomach and go into the plasma. And the pH of the plasma is 7.4. So it will absorb in a better manner into the plasma condition. And in the plasma condition, the ionic form of this drug will be in the higher concentration. So if we talk about the absorption from the intestine, so its absorption from the intestine will not be affected, because this is in the category of somewhat moderately acidic, or you can say weak acidic drug. So second example I have taken is weak base, that is nitrogepam, having pK value 3.2. So if we see here the concentration of this drug in stomach, then it is more in the ionic form and it is less in the unionic form. So its absorption into the plasma will be more from the stomach, you can see here, and it will be in the non-ionic form in the plasma. So it will better absorb from both intestine as well as from the stomach. So if these drugs are both weak acid and weak bases, then these drugs will be absorbed better from stomach as well as intestine when these are in non-ionic form. And if these are in ionic form, these drugs will not be absorbed efficiently into the plasma. So the pH partition theory is best suited for weak acid and weak bases, and it says that weak acid and weak bases are better absorbed from both or entire length of the gastrointestinal tract into the blood plasma. Now, beside the dissociation constant of pK, the total aqueous solubility of an ionized drug is an important factor in the passive absorption of the drug. So total aqueous solubility defined as the sum of concentration of ionized form of the drug as well as unionized form of the drug in solution. So the solubility of unionized form of drug is also known as the intrinsic solubility. So as far as the intrinsic solubility is concerned, it is related with the solubility of the unionized form of the drug. But the total aqueous solubility of the drug is known as solubility of the ionized form of the drug as well as unionized form of the drug into the solution. So for weak acid, ST, that means the total aqueous solubility of the drug will be equal to the intrinsic solubility of the acid in bracket 1 plus 10 to the power pH minus pK. And for weak bases will be equal to the solubility of weak bases in bracket 1 plus 10 to the power pK minus pH. So pH solubility profile for a free acid or free base of weakly acid and weakly base is given in these equations. Now this is the pH solubility curve. You can see here it is between the solubility of the drug and pH. You can see that for free acids the solubility is being increased as the pH is being increased, and you can see in the case of free bases the solubility is being decreased as the pH of the free bases is being increased. So the pH solubility curve is plotted between the weakly acidic and weakly basic drug. So certain generalization can be made on this solubility curve. For weakly acidic drug, when the pH is more than pK, for weakly acidic drug, when the pH is more than pK, then the total solubility of the drug will be much more than the intrinsic solubility, because ionization of the drug increases tremendously. Okay. Now in the second case, when the pH will be equal to pK, then the total solubility of the drug will be equal to the 2 into the intrinsic solubility, because the drug is 50% ionized and 50% unionized. That is why ST will be equal to 2 SA. Now in the third case, when the pH is less than pK, then ST will approximately will be equal to the SA or intrinsic solubility of the drug, since the drug exists predominantly in the unionized form. Now I talk about the weakly basic drugs. Here, when the pH will be more than the pK value, then ST will be equal to SP, since the drug exists predominantly in unionized form. When the pH will be equal to pK, then ST will be equal to 2SP, because some 50% of the drug will be in ionized form and 50% will be in unionized form. And when the pH is less than pK, then ST will be much higher than the SP. ST is the total solubility of the drug into the solution, and SP is the intrinsic solubility of base, because ionization of drug increases tremendously in this case. Now, lipophilicity and drug absorption. We will see the relationship of lipophilicity and drug absorption in this slide. As we all know that pK of a drug determines the degree of ionization at a particular pH. We know very well about this condition. Now the unionized drug, if sufficiently lipid soluble, will be absorbed into the systemic circulation efficiently. And if the drug is in unionized form and having low lipid solubility, then it will not be absorbed efficiently into the systemic circulation. A drug should be in sufficiently aqueous and lipid soluble for better absorption into the systemic circulation. These are the main conditions for better bioavailability of the drug, and this will help facilitate the drug partitioning between a lipidal biomembrane and systemic circulation, obviously. So both aqueous as well as lipid solubility are required for better absorption of the drug. A perfect hydrophilic lipophilic balance should be maintained in the drug structure to become a bioavailable drug. and if the drug is highly lipophilic, then it will not be absorbed efficiently into the stomach circulation. So both are the main constant as rate limiting step. So the drug, if the drug is having good solubility and having the log P value within the range of 1, 2, 4 or 5, so it will be absorbed efficiently into this circulation. Now, the lipid solubility of drug is known as log P, as we all know, and this value tells about the degree of distribution of drug between lipophilic solvent and the aqueous phase of that is water or suitable buffer. So in general, the drug log P value should be in the range of 1 to 2 is sufficient for passive diffusion or passive absorption of the drug. If the log P value is between 1 to 2, then it will be efficiently absorbed into the stomach circulation. So I have already told you that the suitable range is 1 to 4. If the drug is having a suitable solubility, aqueous solubility, then it will be absorbed efficiently into the stomach circulation. Now, a direct correlation between a drug's log P and extent of absorption is illustrated in this table. You can see here, this is the comparison between the intestinal absorption of some drugs through rat intestine and K O by W of the ionized form of the drug. So we will see this correlation in this table that if K O by W ratio all is to water partition coefficient, the partition coefficient of the drug is high here in this case, so absorption is very much high, rapid rate of absorption you can see here. A moderate rate of absorption you will see if the drugs are having a log P value, or you can say the partition coefficient, the moderate region. If the partition coefficient of some drugs are very less, then the absorption of those drugs will be very less, you can see here. So these type of drugs will not be absorbed efficiently into the stomach circulation. Now, for ionizable drugs where the ionized species are responsible for partition, the apparent partition coefficient can be calculated from the following equations. So for log P calculation, some equations are also given for weak acid and weak bases, and you can see in the case of weak acid, log D, that is apparent partition coefficient of the drug, that can be calculated and log D equal to log P minus log 1 plus 10 to the power pH minus pK, and in the case of weak bases, log D equal to log P minus log 1 plus 10 to the power pK minus pH. So I think you got the point related to the pH partition theory, and I hope that I have conveyed the message to you. So as I have already told you in my previous slides that this theory has some limitations also, because it fixes some points, and then it explains that the pH affects the drug absorption. So therefore, in my next video, I'll tell you about the limitations related to the pH partition theory, and then you'll be able to understand that what I'm saying here. So keep watching us, and just do follow my the following videos also to get some more knowledge about these type of theories. So this is all about the pH partition theory. So keep watching us. Thanks for watching.